Cytogenetics, Module 1, Lecture 5, in the Genetics series. Chromosomes were first identified in 1959. Humans have 46 chromosomes. They're in pairs. Chromosomes are actually very tightly wound bundles of DNA. Inside of all the cells in our body, we have our chromosomes, and chromosomes, as I said, are very tightly wound DNA, and the DNA codes for protein. So there are inherited chromosomal abnormalities and acquired. This lecture is primarily going to talk about inherited chromosomal anomalies. When we talk about cancer cytogenetics, we will be discussing acquired chromosomal abnormalities. So indication for doing cytogenetic analysis includes prenatal diagnosis, a woman with advanced maternal age, or a high screening risk for chromosomal abnormality, women with multiple pregnancy losses, or a family history of mental retardation, birth defects, or chromosomal abnormalities. In the pediatric population, children with developmental delay and multiple birth defects. And then finally, cancer-acquired chromosomal abnormalities. The tissues that can be used include blood cells. Blood cells usually don't have nucleus, so it's T-cell lymphocytes, skin, muscle, placenta, products of conception, which is basically everything that comes out when a woman loses a pregnancy. And then tumors and amniotic cells. Amniotic cells are actually skin cells from the amniotic fluid that's sloughed off, and so those are the cells that can be used to do chromosome analysis. And chorionic villi sampling is a piece of placenta. So this looks like a chromosome spread. Here is a cell that didn't pop open, and here are the chromosomes, and they randomly spread. And a karyotype is basically putting these chromosomes in size order. The largest human chromosome is 1. The smallest is actually 21, but they'd already called Down syndrome trisomy 21, so they didn't switch these two. The size of the chromosome doesn't tell us how much genetic material is there, however. About 60% of our chromosomes is non-coding. The chromosome with the largest amount of DNA is 1, followed by 2, followed by 3, followed by 19. Females have 2 X's, so they have 23 chromosomes that look exactly the same. Males have an X and a Y. The Y chromosome determines the male sex. So let's talk about the anatomy of the chromosome. The centromere is the middle, the ends of the chromosome is known as a telomere. The short arm is also known as a petite or P, and Q is the next letter of the alphabet. So the short arm is the P arm, and the long arm is the Q arm. So nomenclature. If there is an addition of a chromosome, for instance in Down syndrome, instead of 46, it'll say 47, XX or XY, and then it'll say which chromosome is extra. If there's a deletion, for instance, if there's a visible deletion on the short arm of chromosome 5, if it says 5p14.2, 5, here's the p14.2, if it just has one line, that means everything above here is deleted. If there are 2 breakpoints, it's an interstitial deletion. That means in the middle. So deletion 5, P1.1.2 to P1.2.3. So this region is deleted. So when you see 2 breakpoints, that means there's an interstitial deletion. We've realized that there are far more interstitial deletions than we originally thought of. The gene location is actually the same nomenclature used to determine deletions are also used to find genes. So, for instance, the cystic fibrosis gene or the CFTR gene is located on the long arm of chromosome 7, 7q31.3. And that codes for the gene that codes for the chloride channel that's abnormal in the cystic fibrosis. Aneuploidy is the most common cause of pregnancy loss. Approximately 50% of all first trimester pregnancies are lost. 1 in 50 of women over the age of 35, and live births with chromosomal abnormalities about 160. When you look at the first trimester losses, the vast majority are numerical, like caused by non-disjunction, and about 4% are unbalanced chromosome rearrangements. When you get to women over 35, it drops from 96 to 85%. In live births, only 60% are numerical, a whole extra missing chromosome. 30% are actually balanced rearrangements that usually don't have a phenotype, and 10% are unbalanced. So of that, in the live ones with a chromosomal abnormality, so 70% have a phenotype, 30% are balanced, so they probably do not have a phenotype. So chromosomal aneuploidies, the common ones include trisomy 21, trisomy 18, trisomy 13, 45X, and 47XXY, as well as deletions on the short arm of 5 and 4. Now normally when we talked about meiosis, cells divide, and it's a reduction division in meiosis. We divide the chromosomes in half, 23 in the egg, 23 in the sperm. The embryo gets 46, divide, 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 and you have a healthy baby. Sometimes mistakes happen though, and if you think about it, there are 23 different places a mistake can happen. So here, let's call this chromosome 1 and 2, they divided evenly into the gametes, but in here, both chromosomes 1 go to one side, and the chromosome 2s divide evenly, so this cell has two copies of chromosome 1 and 1 of 2, and this does not have a copy of chromosome 1 and just 1 of 2. So if this is used for fertilization, we get 3 copies of chromosome 1, 2 from one parent, 1 from the other, and 2 copies of chromosome 2, or hypothetically all the rest. And in this cell, only this parent donates a copy of chromosome 1, and then there's 2 copies of chromosome 2. Monosomy is almost always lethal. The only exception to that is Turner syndrome. And trisomy is usually lethal, and that's why there's such a high risk of pregnancy loss early. So as I stated earlier, 50% of all first trimester pregnancy losses have abnormal chromosomes. Chromosome analysis should be considered in a woman who's had 2 or 3 first trimester losses, or an older fetus with multiple anomalies. It's important to separate the fetal from maternal tissue, so you're not culturing the maternal tissue, and to be certain there should be an equal ratio of males to females in the lab at the end of the year. So here we have, this was a study done back in the 70s of looking at the risk of chromosomal abnormalities, and the risk of Down syndrome increases as you get older. It really starts taking off in the late 40s. So Down syndrome, most babies with Down syndrome have this non-disjunction. They have a copy of extra 21 in all cells in the body. About 2 to 4% are mosaic. At about the 16-cell stage, 3 cells become the baby, and the other 13 become the placenta and the membranes and whatnot. If the pregnancy started with 47XX plus 21, but the extra chromosome was lost at that early stage, and then some of the babies might have the normal amount of chromosomes, and some, if that extra copy of 21 was lost, don't. But that's a rare complication, and the phenotype can be very similar depending on the percentage. Translocation is when one chromosome is attached on another, and in this Robertsonian translocations are between acrocentric chromosomes 13, 14, 15, 21, and 22, and that's also a rare form of Down syndrome. It does not change the phenotype in any way. It only changes the recurrence risk. So let's talk a little bit more about a Robertsonian translocation. This is an individual that has 46 chromosomes, but we have 2 copies of 21, and the third copy is on top of chromosome 14. And here we have an individual that's a carrier. They only have 45 chromosomes, but 1 copy of 21 here, and here's the other. So normally we have 46 chromosomes, but this is an exception to that because there's a Robertsonian translocation. Approximately 1% of the population carries this. So what is the real problem? Well, when we have a Robertsonian translocation, when we're lining up for synapses during meiosis, it's not that easy to do. So the 14s line up, and the 21 tries to line up on the other side of this 21. Now, if the 14-21 goes to one side, and the 14 and the 21 go to the other, then we either get normal gametes or a balanced carrier. However, if the 1421 migrates with the 21, and that's fertilized. We get 3 copies of 21 and 2 and 14. And in this cell, we get, when it's fertilized with a normal individual, 1 copy of 21 and 2 of 14. Here, when the 1421 migrates with a 14, we can have trisomy 14 or monosomy 14. Monosomy 14 is lethal. Trisomy 14 is lethal. Monosomy 21 is lethal. The only of these possibilities that's not lethal is trisomy 21. So in the real world, when an individual has a 1421 translocation, the chance of having a baby with Down syndrome is 15%, because the rest of these are not compatible with life. So here we have normal segregation. So chromosome 14, chromosome 21, and they each go to separate cells. But when we have a 1421, here we have, this is adjacent, but if we have alternate, if the 1421 goes with the 21, we can end up with 2 copies of 21 in the cell. So, if a parent carries a 1421 translocation, there's a 5 to 15% chance of having a baby with Down syndrome. But if the baby is born with Down syndrome and has a 1421 translocation, there's only a 50% chance that the parent carries it. The parent may not carry it. It may have been something new in the egg or sperm. So testing the parents is important. A baby is born with features consistent with Down syndrome. What is the most likely chromosomal analysis? 47XX plus 13, 47XX plus 21, 47XX plus 21, 46XX, 46XX translocation, 1421 plus 21, or 47XX plus 18. And this concludes this module on cytogenetics.